All right, so most of you, I'm sure, here are familiar with northern pike as an invasive species, but I'm going to start by just providing a little bit of background. So northern pike are broadly distributed across the northern hemisphere. As you can see on this map in blue, their native range is represented, and it's quite broad. And where they occur, they prefer shallow waters with emergent vegetation. And that's because they are ambush predators, so they hide in that emergent vegetation and swallow up their prey, basically ambush them. And they're very effective at this. Where they occur, they're also considered top predators, and they can consume anything up to 75% of their body size. This map shows their distribution in the U.S. The yellow areas represent locations where northern pike are native, and as you can see in the pink and red areas that represent places where they've been introduced, these fish are quite prominent outside of their native range. They've been largely introduced as a sport fishery, yet where they've been introduced, there are rising concerns on the impacts that northern pike have on the native communities, and that's because there have been a number of studies documenting the declines that those native communities have suffered as a result of northern pike invasions, to the point where we've even seen extirpation of populations of important native species like salmon. This map is zooming in on my study area, which is the upper Columbia River Basin, and that is spanning from western Montana over into eastern Washington. And I'm going to use this map as a little bit of a timeline to show you what we know about the history of northern pike in this region. I also want to acknowledge that this map was originally put together by some of my collaborators at the Kalispel Tribe of Indians and the Confederated Colville Tribe, and we've been able to modify this as we've chatted more with biologists in Montana and other regions to get a better sense of this history. So the first known occurrence of northern pike west of the Rocky Mountains was in Lone Pine Reservoir, and that was a transport dating back to 1953, where fish were taken from, I have no pointer. Fish were taken from, I'll try and point right here, East Glacier, Montana, where there is a small population of native northern pike in that region, and were transported to this reservoir in western Montana. From there, we saw northern pike emerge in the Flathead River Basin and also in the Bitterroot River Basin in the late 1960s and 1970s. We then saw them spread into the Clark Fork River Basin, and we also know there was another independent introduction of northern pike in the 1970s in the Coeur d'Alene River Basin. From there, those northern pike spread downstream into the upper Spokane River by the late 1970s. There was yet another introduction of northern pike in the 1980s in the Clearwater River Basin, and in 1997 and the late 90s, it's believed that high flows associated with high snowpack led to downstream drift of northern pike, both from the Clearwater River Basin into the Blackfoot River Basin near Missoula, as well as into Lake Pend Oreille. From there, we didn't see a whole lot of change until about 2004, northern pike showed up in the Pend Oreille River. And at this point, we saw the pike expanding rather rapidly. They were established 5 years later in the Columbia River in Canada, and then showed up downstream in Lake Roosevelt. Much of my talk is going to focus on Lake Roosevelt, and I want to point out that this lake is actually a reservoir within the Columbia River that extends basically from the border of Canada down to Grand Coulee Dam right here. I'll do that also on this slide here, down to about there. And at that point, managers became very concerned because beyond Grand Coulee Dam, we have ESA-listed stocks of salmon and steelhead. So around 2012, or in fact in 2012, the Kalispel Tribe of Indians began a very aggressive suppression effort with gill nets to remove northern pike from the Pend Oreille River, and that has been largely successful. In the last 8 years, they've been able to reduce northern pike numbers by over 90%. Yet we still have this situation where we have northern pike continuing to expand downstream because the population downstream is still established. Now, from a management perspective, we have a couple different information needs in this instance. We first want to be able to monitor the leading edge of that invasion to detect any changes and to be able to respond with suppression efforts in a very focused manner. Yet in areas where we've been successful at removing northern pike, we also want to ensure that we're not seeing repeated introductions so our efforts aren't totally in vain. So with that in mind, this is a northern pike from Lake Roosevelt. It's a 26-pound female. That's a pretty big fish. And if it can eat anything that it can fit in its mouth, which is usually how northern pike work, if it fits in their mouth, they'll eat it. I think all of our native species are pretty much at risk, at least at some life stage at some point. So with this issue, we have some areas where we've been successful at really suppressing northern pike, and other areas where they continue to expand. We really have two major information needs. One is to monitor the invasion for any early detection of expanding populations, and the second is to identify the sources of those invading those areas to prevent repeated invasions. So I'm not going to actually talk very much about that first point, monitoring for early detection, but I do want to mention that I have been involved in some work with the Confederated Colville tribe using eDNA to monitor the expansion of northern pike in Lake Roosevelt. We published on this recently in the journal eDNA, so you can look there if you're interested, or come find me, and I'd be happy to chat with you more about that later today. I do want to highlight that we've been using eDNA eDNA was applied here largely to supplement ongoing creel surveys and gillnet efforts for northern pike in this area. It's been very useful at helping us detect small-scale changes before we see live pike using those other methods. It's also been useful for areas where creel surveys and gillnets are just not feasible or easily applied. But what I really want to talk to you today is about identifying the sources of these invading northern pike in eastern Washington. And to really think about this from kind of a scientific perspective, the scientific question we're asking is, where did these pike in recently invaded areas come from? To address this question, we've collected tissue samples from populations, 18 populations across our study system. The green circles up here in eastern Washington are the areas that are considered recently invaded populations, and all of the gray dots represent samples from established populations upstream. And just broadly across this landscape, what I want to highlight is this is a relatively connected landscape with some fragmentation. We have two major river basins that we're considering, the Clark Fork River Basin and the Coeur d'Alene River Basin. And this whole river basin is kind of stippled with a lot of dams. So the dams are represented here in yellow arrows. They point in the downstream direction. And this is really important because it would be relatively easy, for example, for a fish from the Clark Fork to swim down over these dams and end up in eastern Washington on its own volition. So downstream volitional spread of those northern pike. In contrast, if a fish from the Coeur d'Alene River Basin were to get up into eastern Washington, that would likely need to be aided by human transport, because we know that pike, while they may be able to swim down these dams, they cannot swim upstream over them. All right, so our first hypothesis from a genetic perspective answering this question is, if northern pike were simply the product, northern pike in eastern Washington are the product of downstream drift, we would expect to see what geneticists call isolation by distance. And what that means is that the populations in the eastern Washington would be most closely related to the nearest upstream population. And in this case, that would be Lake Pend Oreille. In contrast, if the fish in eastern Washington are the product of human transport, then what we would expect is the populations to be related to one of these individual populations, but that might not necessarily be the nearest neighboring population. So it could be basically any one of these 18 source populations. All right, so our methods. As I mentioned, we sampled 18 populations and had over 450 fish in our data set. We estimated levels of genetic diversity. I should also mention we used 11 polymorphic microsatellite loci to analyze these samples, and that was on a panel that was developed at the Minnesota Department of Natural Resources. We then used that genetic information to look at levels of genetic diversity. We looked at relatedness between the populations, so genetic diversity at the population level and how they relate to each other. And then we also performed a genetic assignment analysis, looking at individuals in recently invaded areas and how closely related that each individual may be to each potential source population. All right, so here are our results. This table lists all of our populations in the study. In gray, you see the potential source populations, and in white or unshaded are our recently invaded areas. And basically what this is showing you is N, the sample size, and then HE is heterozygosity, a measure of genetic diversity, and allelic richness, which is a measure of genetic diversity that is corrected for sample size. And I've sorted these populations based on heterozygosity, and you can see that those populations in the recently invaded areas have the lowest levels of heterozygosity. Now that's telling us a couple things. One, that those recently invaded areas were likely founded by a subset of individuals, and two, that we're not seeing a constant influx of individuals from the source population. If we were seeing a constant influx, we would expect the levels of heterozygosity to rise very quickly after the invasion and start to look much more like that of one of these source populations. All right, so the next question was, how are these populations related to one another? To look at this, we performed a principal coordinates analysis. For those of you who aren't familiar with this, the general summary of how this works is we take all of the genetic information represented in each population and condense that down to 2 coordinates that we can then plot on a graph and just visually look at how the populations are clustering on that graph. And this is what that looks like for our data set. I've color-coded these based on their drainage of origin. So we have our populations in green, which represent the recently invaded areas. And you can see that those are really sticking out on their own away from the other potential source populations. Now, again, that's not entirely surprising because we know these individuals were founded by some subset where they're still with the lower levels of genetic diversity. We are still seeing what geneticists call founder effects, but it's still useful to see that these are falling out here. Another thing that I think is useful to note is if you recall, the source of fish in Lake Pend Oreille was hypothesized to be downstream drift from the Clark Fork River Basin. And that looks like that is largely supported in this data set. So these fish in Lake Pend Oreille are closely related to those that are right upstream. They cluster together. So it looks like that these data are supporting that that was the origin of fish in Lake Pend Oreille. They were washed downstream by high flow events. Yet this still does not answer our question, where exactly did these fish come from, right? These fish are diverged because they have lower levels of genetic diversity currently. And so to address this question, we performed a genetic assignment analysis. This is what that looks like on the landscape. The color coding has been reserved here. So we have our fish in the Clark Fork Basin, our fish in populations in the Coeur d'Alene, our recently invaded areas, and then a few populations that we're considering relatively isolated without connection to these other areas. And so what we're really asking with this assignment analysis is for each individual in a recently invaded area, what's the probability that it came from each one of these source populations? So what's the probability, for example, that a fish came from Long Lake or came from Cabinet Gorge? And what's the probability it came from Thompson Falls? We looked and compared each fish to each potential source population. All right, we did this in Gene Class 2, and in this software, we did not allow the individuals to assign to their location of capture. That's because we know reproduction is going on in Lake Roosevelt and in the Pend Oreille River and in that area, and so if we allowed them to assign to their location of capture, it's likely that they would just assign back to where they were captured. But what we really want to know is where did they come from? Where did these populations generally come from? So we didn't allow them to assign to their source. We forced them to assign to an outside source. Secondly, we did not force assignment. So we did not say that you have to assign to one of these populations and all of these populations are the only populations you could have come from. If the probability of assignment was unlikely, it's possible that some fish could assign with a 0% probability to any single source population. All right, and this is what we found. By and large, nearly all of the individuals had the highest probability of assignment to Cave and Medicine Lakes, which is in the Coeur d'Alene River Basin. And really what that tells us is that our second hypothesis is supported. It's most likely that these fish in eastern Washington did not wash downstream from the Clark Fork River Basin, but were transported by humans. But there are still some nuances to understand here. Although cave and medicine lakes were the most likely source of assignment, the variation was huge across our 145 individuals. So some individuals assigned to this location most strongly, but their probability of assigning to cave and medicine lakes was less than 1%. We had some individuals that also assigned there with near 100% probability. And when I was thinking about how to represent this information, it's a lot to try and compress down 18 populations by 145 individuals. And so I thought that a heat map might really help with this. And so what you have here is each individual is represented by a row, and we have 145 rows, and then each source population is represented by a column. And the cell that corresponds to that individual and a potential source population has a probability of assignment that is represented by a heat scale. So the more bright, the more white hot, the higher that probability is. This is how I've listed our individuals, starting from upstream and going to downstream as Lake Roosevelt is at the leading edge of the invasion. And then we have our source populations across the bottom. And what you can see here is that, again, cave and medicine lakes assign with a very hot, high probability, but we still have some individuals like this individual here from Lake Roosevelt that while it assigns in our data set to Cave Lake Medicine Lake with the highest probability, that probability to assigning to that location is actually relatively low, and it's, low for all of the other populations. And so what this is telling us is that we may have source populations that contributed to this invasion that are not represented in our data set. So our conclusions, the results suggest that pike in eastern Washington were introduced illegally by human transport, and that multiple sources may have contributed to this invasion. Low genetic diversity suggests that recently invaded areas are not receiving a constant influx of fish. But what's not surprising and what is reinforced by these data is we are likely managing people more than we're managing fish alone. And so thinking about management from the perspective of communicating the importance of not moving invasive species and the impacts that pike can have in areas when they are reintroduced is going to be really important in this context. And that is all I have for you. I want to acknowledge some of my partners, especially those, my collaborators at the Confederated Colville Tribe and also others at the Kalispel Tribe of Indians that have been especially helpful in all of this research. And with that, I will take any questions if there's time.